Good evening. I'm going to go ahead and get started because I'm going to go over a few introductory remarks before we have our speaker for the evening. But before I do that, I just want to thank you all for coming today. I know it's like, oh, should I just go home and have dinner or should I go to the Stem Cell Center? And thank you for making the decision to come here and be with us this evening. We very much appreciate it. So I'm going to do just a few introductory comments about the Stem Cell Center. So before I get too deep into that, I just want to say thank you to the people who organize these events, these community lectures. It's actually not me. Oh, and by the way, my name is Lisa Flanagan. I'm one of the faculty here in the center. I'm in the Department of Neurology. And I have a joint appointment in biomedical engineering. But I don't organize this. This is organized by Brian Cummings. He's our community outreach chair. And then Abby here in the center is just a huge force to make sure that this whole thing happens. And then UCI Media, we thank them as well because they come and help us out with this when we do these community lectures. So I want to talk just a little bit about, how the science goes, especially for like stem cell science and things that we think about that we want to, move into translational realms. How does that kind of happen? And it all is sort of these advances that we think of that end up helping human health. They start with places like UCI, with universities like UCI, and groups of scientists like we have here in the Stem Cell Center. So if we think about how much impact the research that happens at universities has on, development of new therapeutics and things like that, we can just look at these numbers. This is over a particular year. There were so many patent applications and patents that were awarded based on research that came out of universities. So it's really a driving force to take some of these basic science findings and turn them into products or therapies or things that will impact human health. So that happens here in our center. Oops, I think I went too far. This is just, well, anyway. I don't know why that's doing that. These were not my slides, so I'm just going to do that. If you look really quickly, you can see these various companies that have come out of research that is here in the center. So, they're secretive. We don't want to show them for too long. But anyway, we're really happy that there's that kind of pipeline moving from these basic studies into products that can help people. And so if we, there are certain things that we have built into the center that help to make that possible. And one of those is our Alpha Clinic for Cell and Gene Therapy. And that's run by Dr. Daniela Boda. And this group has made such a difference in terms of how we're able to take our basic science discoveries, bring them into the initial clinical trials to check for safety, and then the next parts of the clinical trials where we check for efficacy. So is this particular treatment having an impact on the disease? And then finally, we hope that these get incorporated into standard of care to treat various diseases and injuries that affect people. And so there are a lot of trials through our Alpha Clinic, and it's been growing. So I think that's really, for me, that's really astonishing how much it's grown since 2019, and this graph goes through 2022. So we've had a big increase in the clinical trials that are being run through the Alpha Clinic here in this building. So right now we have more than 20 disease indications that are being addressed by clinical trials. We have some critical partnerships, and particularly with the Long Beach VA, the Veterans Administration there, Children's Hospital of Orange County, and then University of California, Riverside, their health system. They're all part of our group here for this Alpha Clinic, and that's been a really important group of strategic partnerships for us. So the other thing that we're really excited about is our new GMP facility. So that's GMP stands for Good Manufacturing Practice. And the key thing when we think about that is, okay, so we're scientists and we're working in our labs and we're doing something with cells, but if you think about how do we move that out of the lab into like clinical practice, there's a lot of steps that have to happen in between. And one of the key steps is having a way to manufacture the cells so that there's enough to run a clinical trial. I mean, when we're doing them in our labs in a dish, it's just this tiny little grouping of cells. But if you think about some sort of a stem cell therapy or other therapy for people, you have to have enough cells for the clinical trials, which could be run not only here, but across the rest of the United States. So how are you going to have that large group of cells, and how are you going to make sure the cells are good and are, having the characteristics that you need them to have to treat that disease process? And so one of the key things then is to have a facility that can manufacture this large amount of cells. And so you can see some pictures on the left. We have this new facility, and it's run by Roberta Marino, who is our new manager of this. And you see the person in the hazmat suit. That's to make sure that there's no cross-contamination of cells as they're preparing the different groups of cells in different rooms. But we're really super excited about this, and we're actually going to have a grand opening in a couple of weeks on May 22nd. That will be our grand opening for this new GMP facility. So lots of exciting things are happening here, and we're glad to be able to share them with you. And so now I'm going to do a quick introduction for our speaker for tonight. I'm really happy to introduce Asuka Eguchi. She's an assistant professor in the Physiology and Biophysics department here at UCI. Her lab focuses on muscle diseases, and particularly heart and cardiac muscle. And as well as that, she also focuses on neuromuscular disorders and the use of gene therapy to correct muscle defects. She uses stem cells to study human disease to try to identify new therapeutic options. Before coming to UCI, she completed her Ph.D. training at the University of Wisconsin-Madison, which is really kind of a stem cell hub. There's a lot of great scientists there working in the stem cell space. So I think that was probably a great place to get wonderful foundational knowledge in stem cell biology. She then moved on to do a postdoctoral fellowship at Stanford University with Helen Blau, who's a luminary in the field of muscle research. So that's also a fantastic place to do training. She has numerous publications, and in addition to that, she has 2 patents. So she's already, on the way to moving her research, like we were talking about earlier, from some of the basic science studies to something that could eventually be applied to human health. As a new assistant professor, she's already garnered significant funding for her research program, including a prestigious NIH K01 Career Development Award. She's delivered scientific lectures across the United States, and I'm really happy that we have a chance to hear her presentation today. Thanks.
Okay, thank you, Dr. Flanagan, for that very kind introduction. And I am excited to talk to you today about gene therapy strategies for muscle diseases. And so here are some examples of muscle diseases, and highlighted here in black are examples of muscle diseases where there are genetic mutations that directly affect the muscle. And then there are types that affect the nerves that innervate the muscle, and those are highlighted here in red. And then sometimes there are types of diseases caused by enzymes that help the muscle function properly. And all of these diseases are not necessarily caused by the lack of a protein being expressed. Sometimes it's caused by, for example, in the case of FSHD, there's too much protein being expressed. And so today I will be focusing on gene therapy, which is a replacement transfer therapy. So these 4 disorders here have clinical trials that are ongoing to replace the gene that is mutated in these patients and then restore the protein, or at least the protein's function for these diseases. So what is gene therapy? It is often delivered by adeno-associated virus, and so AAV is it does not cause disease in humans, but we can use it as a delivery mechanism to deliver genes to patients. And there are three components to it. One is the vehicle. So this is the capsid that encapsulates the payload. And here you have the promoter that determines where the gene will be expressed. So where the protein will eventually end up. And so we want to target certain tissues, not all organs, and so this is how you control that gene expression. And then the gene is actually going to encode for the protein that might be missing in patients. And today I will be addressing some challenges in gene therapy, and then I'll be discussing more in depth about a specific disease called Duchenne muscular dystrophy. And then I'll go into how we use stem cells to model disease and test therapeutic strategies. So first, the challenges. And so the major challenge is that adeno-associated virus, AAV, has a packaging limit. And so you can encapsulate this part of the gene or the payload that has the viral elements, the promoter, and the gene. And all of that has to be less than 4,700 base pairs. And for some genes, that's not big enough. And I'll be talking about an example of that later on. But the packaging limit is one of the major limitations for gene therapy. But the nice thing about gene therapy using AAV is that it is effective in targeting some of the tissues that we are interested in delivering a protein. So that's the skeletal muscle, diaphragm, and the heart. And the diaphragm helps you breathe, and then the heart helps you pump blood, and all of these are muscles. So the other major challenge is that when AAV is systemically delivered so that it can target all of the skeletal muscle, diaphragm, and the heart, a lot of it, 80 to 90% of it, ends up in the liver. And you can see here that this is on a log scale, and there's not much of a difference between male and females, but there's 10 to the 6 viral particles in the liver compared to other tissues that get at least an order of magnitude lower than that. And so this is definitely a major challenge because now in clinical trials, in order to target all the tissues, patients are getting 10 to the 14 viral copies per kilogram. And so that amounts to be a huge amount of virus that makes it very challenging to produce and also toxic to the patients. And I just mentioned that AAV is used as a vehicle because it's not toxic, but anything in very high quantities can be very toxic. And so 10 to the 14 is really bordering on the edge of ability to deliver to the muscle, but at the same time causing liver enzyme problems and even death in some cases. So it is still a major hurdle to overcome. But then there was a major breakthrough recently where in this paper they screened through many types of capsids and found that these RGD, so arginine, glycine, aspartic acid containing residues can help uptake in the muscle and reduce the delivery to the liver. And so this definitely is an avenue that people are exploring to try to implement this strategy to lower the dose required. And then the other challenge is in seroprevalence. So in the natural environment, there are AAVs, and we get exposed to them. And so here, this is showing that when children are very young, 2 to 7 years of age in the black bars, there is already anywhere from 5 to 30% of individuals have neutralizing antibodies against AAV. And these numbers on the bottom here refer to different capsid types. And so there are certain capsids that target the muscle better, and AV8, AV9, and RH74 are the main ones that are being used for neuromuscular disorders. But then by the age of 8, a lot of the population already have developed neutralizing antibodies, which makes them ineligible for gene therapy. And so in order for you to get a gene therapy, they first take a sample of your blood, and then they determine whether you have neutralizing antibodies specifically for the capsid that they are using for the therapy. And if you do, then you are not eligible for therapy. And then on top of that, this is a one and done type of strategy. So if you get AAV gene therapy, that means that you are no longer eligible for AAV gene therapy in the future. Although now there are efforts to basically clear the neutralizing antibodies from the blood in order to be able to redose patients. And so those efforts are largely being led by pharmaceutical companies. And the other major challenge is that in order to make AAV, you have to use ultracentrifugation methods. And these are very high centrifugation methods. And this is in order to make sure that the capsids have DNA inside them. And this is a slide that was lifted out of Solid Biosciences presentation in 2021 after the FDA asked them to clarify their production methods because some of their patients were getting toxic liver reactions to the gene therapy. And they found that their manufacturing process basically had half of the capsids did not have any DNA, any therapeutic component in them. And so now they've improved their manufacturing process, and they can get a higher number of capsids that actually have the therapeutic component. But these methods to actually produce AAV at such large quantities, basically 10 to the 15 viral particles per patient, that makes it extraordinarily challenging. And currently, the FDA has approved 2 gene therapies, and one is for SMA, and the other is for Duchenne muscular dystrophy, and it's for a subset of patients, infants in the case of SMA, and for Duchenne, it's 4 to 5-year-olds. But the price tags are very high, partly because the manufacturing process is so challenging. And in summary, there is a limitation in the packaging, and then it's still difficult to target all the tissue types that we're interested in, the skeletal muscle, diaphragm, and the heart. And then not everyone is eligible for gene therapy. If you have previously been exposed to AAV, you would not even know it because it does not cause disease. And you just have to get tested for neutralizing antibodies. And then also, as Dr. Flanagan mentioned, you have to have good manufacturing practices, and this process of making AAV is not easily scalable. And Now I'm going to talk specifically about a disease that I'm studying, Duchenne muscular dystrophy. So this disease is caused by the lack of dystrophin expression, and patients experience severe muscle weakness, and it's progressive. And this lack of dystrophin eventually leads to boys being in wheelchairs by their teenage years. And then here you can see the muscle biopsies from a healthy individual versus a Duchenne patient. And you can see a lot of white, which is the connective tissue, the scars in the muscle. And then you can also see that there's a lot of variation in the size of the fibers. And in addition to the skeletal muscle dysfunction, these patients also have respiratory problems, and they also have problems with their heart. And so currently, patients are taking corticosteroids because they have this process of constantly degenerating their muscle and then also regenerating their muscle. And this constant process causes immune cells to clear all the debris that is necessary to make new muscle. And because they're constantly in a state of inflammation, they're taking corticosteroids. And they also, because of their muscle weakness, tend to have scoliosis, and so that is what they need surgery for that. And then they also have these eccentric muscle contractions that they have physical therapy for. And then they also have assisted ventilation devices. So this disease is caused by the lack of dystrophin expression, and this gene is located on the X chromosome, which means that it's a disease that affects boys. And without this protein, this basically acts as a shock absorber, and it transduces the force from inside the cell to the outside of the cell. And without dystrophin, you also get a lot of holes in the membrane, the cell outside of the cell, or the barrier of the cell. And this makes it very difficult for ions that help the muscle contract to be regulated properly. And mutations in this gene, this is the largest gene in the genome, and so just by sheer probability, there are going to be mutations. And a lot of the mutations occur in this region of the gene, although this is N-terminus can also be a hot spot. And all types of mutations can occur, deletions, duplications, rearrangements where there are parts that are flipped around. And there are a variety of means to try to restore gene expression when the patient expresses no protein. And then there are individuals who have mutations that render an expression of a protein, but it's shorter. And so today I'll be talking about gene replacement therapy, where in the Duchenne patients that express no protein, where this strategy would help. So one of the, so really this is diagnosed by sequencing the gene, and usually parents consult a neurologist if there are delays in developmental milestones, and then the first thing to do is to check for creatine kinase levels. And this marker in the blood basically tells you if you have a lot of muscle degeneration. So after a patient is diagnosed with Duchenne, then every year the patient undergoes a functional test called the North Star Ambulatory Assessment Test. And this basically assesses the ability for the patient to use their legs, but also parts of it involve their upper limb muscles. And you can see that this is a very simple scoring scheme where it's 2 points for successful, 0 for unable to perform, and then 1 for some needing assistance or struggling with the task. And it's made to be a very simple test so that different people who make the assessment can score an individual in the exact same way. And one of the classical signs of Duchenne patients is the Gowers maneuver. And this is a test where a patient from a lying position stands up. And in order to stand up, they have to first adopt this tripod position and then push their hands against their thighs in order to achieve this standing position. And if earlier in the disease they are able to stand up with no problem, and then by the time they're in wheelchairs, it is very difficult to perform the task. And there are very different trajectories on how the NSAA score declines. And this is an example of all of these patients that were tracked over time. And then, but from this information, they were categorized into different categories. You can see here in red is a progressive decline from the age of 4. But most patients actually improve their NSAA score from the age of 4 to 7, and then there's a decline. And so a lot of the clinical trials are using the NSAA score as a primary endpoint. And so the heterogeneity, in addition to the fact that young patients are being targeted for the clinical trials, makes it quite challenging to make assessments on efficacy. Once the patient is in a wheelchair, then they're not able to perform the NSAA test. And so then they, clinicians refer to these performance of the upper limb tests. And these tests basically test whether a patient can move their arms in different directions. And at very late stages of the disease, it's difficult for patients to bring food to their mouth. And so it's testing in various ways, from lifting objects to raising their hands above their heads. Also in performing these mazes where they just track their finger across the sheet of paper. But as you can tell, all of these rely on effort from the patient. So sometimes it can be challenging to get a good score out of a patient if they're not willing to perform the task. So that's another challenge with this. But in terms of current treatments, I mentioned that corticosteroids are a main standard of care. And so with the use of corticosteroids, there's evidence that it preserves their muscle function, and patients who take corticosteroids become reliant on wheelchairs later in life, and then it can extend that period from 1 to 2 years. And then there's also evidence that it helps with their respiratory function and cardiac function. But one of the side effects is that there's weight gain, and then they have osteoporosis. There's also, eventually, corticosteroids, prolonged use does yield to desensitization, and then they're not able to, the corticosteroids don't work anymore. And I also mentioned that due to the muscle weakness, they have scoliosis, so they have these surgeries for that. And so this was the old method to straighten the spine, and then now this pedicle screw fixation is the current method. And in terms of physical therapy, this is basically geared towards preserving their muscle function and also reducing the eccentric contractions. And you can see that there are a lot of stretches, and then also some cycling and swimming, things that are a little bit less taxing on the heart. And then these orthotic devices can also help with patients in lengthening that calf muscle. And previously, patients were mainly dying of respiratory failure, but now with ventilation assist devices, patients are living longer. And this graph here shows essentially that people who were ventilated survived in their 20s, and then now patients are primarily dying of heart failure. But these ventilation assist devices can be more, they come in two different forms, non-invasive, and then where you actually have to have a device attached to you. So now that patients are living longer into their 20s and 30s, the primary cause of death is heart failure. And that is what I'm focusing on. And heart failure is caused by the premature loss of the essential cells in the heart. And this causes infiltration of fibroblasts and fat cells that basically change the structure of the tissue here. And at first, there's this thickening of the tissue, but eventually there's very little muscle left and a lot of scar and fat tissue, and then this wall becomes very thin. And then, you've probably had a scar before, and you know how stiff that feels, and that makes it really hard to for the heart to pump blood out of that left ventricle now. And due to the weakening of this muscle, patients can have a low ejection fraction by echo, and then they can also have arrhythmia. And the resting heart rate is actually high even in the womb. And current treatments basically reduce that mechanical burden on the heart by beta blockers that slow the heart rate, or ACE inhibitors that lower the blood pressure. So these are non-specific drugs that basically make the heart work less. And so I mentioned that dystrophin is the gene that is not expressed in patients. And the inspiration for gene therapy came from this individual. And so this person had a major mutation in dystrophin, was missing 50% of the gene, but was in the Army in his 20s. This was at the age of 45. He climbed a cliff. And then this is him in his 60s walking with a cane. So it became clear that this gene that encodes a 427 kilodalton protein could be quite functional even if 50% of it is missing. And the gene that would, or the coding sequence for this product is still too large to fit into AAV, but it basically inspired researchers to see if there could be other domains of dystrophin that could be taken away so that it can fit into dystrophin or AAV. And so after over 20 years of research, these microdystrophins, these miniature versions of dystrophin, they materialized into clinical trials. And currently, Solid Biosciences, Pfizer, Sarepta Therapeutics, and REGENXBIO have these ongoing clinical trials. And for Sarepta, their gene therapy has been approved for 45-year-olds by accelerated approval, even though there's not a clear indication of therapeutic efficacy yet. And all of this was made because these miniature versions can fit into AV. And in preclinical studies, these miniature versions of dystrophin, they showed efficacy in animal models, specifically in mice and dog models of Duchenne. And so in these clinical trials, first there's this preclinical phase where there's testing in animals. And then in phase 1, the safety is assessed. And in rare disease, this is actually tested in the patient population. Usually phase 1 happens in healthy individuals, but for rare disease, it's actually tested in the patient population. Phase 2 tests efficacy in a small cohort of patients, and then phase 3 is efficacy in a large cohort. And so for rare disease, we're talking about small numbers. This is less than 10 people, less than 100 people, hundreds of people in phase 3. For example, this is in stark contrast with the COVID-19 vaccine, where it was tens of thousands of people for the phase 3. So the next part I'll be talking to you about was really motivated by conversations that were discussed by parents of Duchenne muscular dystrophy patients. And so there's this conference organized by Parent Project Muscular Dystrophy, and these pharmaceutical companies were presenting updates on their clinical trials. And a lot of the parents asked, well, what will happen to my son's heart if they get the therapy? And there was no very clear answer. This is largely because animal models, they do not have the severe cardiac phenotypes that we see in patients. And so now I'll move on to some of my research. And so these mouse models that lack dystrophin, they actually have heart problems at a very old age, over 16 months of age, which is comparable to a geriatric person, whereas in patients, heart problems can be observed even at the age of 10, where there's fibrosis by MRI, and then patients are dying in their 20s and 30s. And then the mouse models also have a mild muscle weakness. They do not have curvature of the spine, and then they live to be a normal lab mouse, living to the age of 2 years. And some of the limitations in learning about the cardiac efficacy is that in the clinical trials, if the patient has any indication of heart problems, they are excluded from the study. And in addition, it can take years for heart problems to develop, and progression is much slower in the heart in terms of functional decline. And so the clinical trials that last 2 to 5 years may not capture this efficacy, especially in a small population. And so I'm using human-induced pluripotent stem cells, where you can convert cells from the blood that are derived from the patient and have the same genetic makeup as the patient, convert them into stem cells, and then now we have all types of recipes to make different cell types. And the cell type I'm interested in is the heart. And these heart cells are really fun to make because they beat spontaneously in a dish as sheets of cells on day 10. And then later, if you keep them in a dish too long like this, then they completely lift off the plate. And so we dissociate them from each other and then replate them. And we're able to test in a dish whether these gene therapy strategies are effective. And today I'll be presenting data from stem cells that are derived from 3 different individuals, and then basically testing. It's a clinical trial in a dish. And so at day 10 of differentiation from stem cells, we introduce either an empty vector, so equivalent of a placebo, or the gene therapy, and it is one of the 3 miniaturized microdystrophins, and then test in these Duchenne cells where the dystrophin mutation exists, and then alongside it are the healthy control cells where the dystrophin mutations are corrected. And then some of the striking phenotypes that we see in a dish is that the cells have this much smaller morphology. So they are a lot smaller, their nuclear size is smaller, and then the staining here in green is one of the sarcomere proteins. It helps the cell contract, and the density of those proteins is lower in Duchenne. And then we can also electrically stimulate the cells. And when we electrically stimulate the cells so that they beat every 1 second, typically what you should see are these really beautiful calcium traces, where there's a nice spike of calcium every 1 second. With Duchenne, you start to see deviations from these 1 second intervals. You also see these double spike peaks that are indicative of arrhythmias. And so we see a higher frequency of these strange or arrhythmic traces. And then also we can measure their force of contraction, and it's lower in Duchenne. And then when we track the survival of the cells over time, then we also see that the Duchenne cells have a shorter lifespan. And so in the comparisons, we are comparing an empty vector control versus one of the microdystrophins, and then comparing whether we can reverse some of the phenotypes and bring them back to healthy control levels. And some of the transgenes, so the genes that are delivered, the unique properties is that this one recruits nitric oxide synthase, this one has an extra spectrin repeat that gets anchored to the plasma membrane, and this one has an extra spectrin repeat that binds the microtubules. So all of them have this N-terminal actin binding domain. They also have this cysteine-rich domain that gets anchored to the plasma membrane, but there are some distinct and unique properties of these genes, and that's where I'm trying to make the comparisons. And just asking whether these different disease characteristics can now be reversed or made a little bit better. And so far, we find that despite expressing the microdystrophins, the cell size remains smaller, and then the cardiac troponin T density is quite variable depending on the microdystrophin, but in one of the cases, it seems to improve it somewhat. And then for the calcium measurements, we capture these different traces. Again, we can capture these early after depolarization events that are indicative of arrhythmias. And largely with the healthy cells, we see a higher incidence of these healthy traces. So when you're looking at these graphs, gray is healthy, and that's what we want to see. With the Duchenne cells, you start to see these more aberrant traces, and then with the delivery of the microdystrophins, you see a bump in that gray bar. And depending on the Duchenne line, you see various outcomes, and there doesn't seem to be any consistent trends among the different comparisons. And then, but despite having some of these arrhythmic problems, they do help with survival in a dish. And so this is a cell viability assay where we compared day 40 and day 30 cells that were viable in a dish, and that the delivery of the microdystrophins helped with the survival of the cells. And this project is still ongoing, and we are continuing to measure whether the microdystrophins can help with the mechanical contraction of the cells. So this is a traction force microscopy, where we cast these hydrogels with green fluorescent microspheres embedded in them. And by tracking the displacement of the microspheres, we can, and then based on the stiffness of the gel, we can calculate parameters of contraction like force, velocity of contraction, velocity of relaxation, and synchronicity. So we hope to learn more about how the microdystrophins are helping. And we also are interested in delivering larger forms of dystrophin. And so there are new strategies to basically deliver two components of AAV, and then have these two independently expressed protein units spontaneously ligate together in the cell. And so we're exploring this strategy. Elaine, sitting back there, is working on that. And then we're also exploring the delivery of full-length dystrophin by mRNA, but mRNA delivery would be a transient approach, so it would not, it would require multiple dosing. So we're actually going to test what type of dosing is required to reverse some of the heart phenotypes. And then we're also using patient data, so trying to correlate the mutational status of a patient with their cardiac function. So this is echocardiograms, ultrasound of the heart, and then low ejection fractions indicate that the heart is functioning poorly. And so if we can make correlations between the mutation and these low ejection fractions, we can maybe infer more information about which domains of dystrophin might be important. And so in summary, microdystrophin gene therapy has shown promise in the skeletal muscle, but little is known about efficacy in the heart. And these human stem cell derived cardiomyocytes, they enable the testing of these therapeutic strategies in the molecular context of human cells. And then our preliminary data shows partial rescue of the calcium handling, and we want to test new design strategies that might enable larger variants to be expressed. And with that, I'd like to thank the people who contributed to these studies and my funding, and I'm happy to answer any questions.
Wow, that was super interesting. Thank you so much. So I have the microphone, so if anybody has questions, I can bring the microphone to you so that our speaker can hear your question. So questions. All right, while you think about your questions, I'm actually going to start with one. So I think that's so interesting to think about how everybody's trying to use viruses to encapsulate the gene that you
Yes.
want to deliver and then get that into the cells. And I was wondering, it's like you kind of mentioned people have focused on AAV as a particular kind of virus that they've used for a long time. But as we get further along with, protein engineering, viral engineering, those kind of things, are people going to try to create like an engineered virus that would be able to have a larger DNA, payload, as you mentioned, so you could get a bigger gene, like the whole dystrophin gene in a virus? Or are there just limitations to what the cell will take up or something like that that make it hard to do that?
So there are definitely people in that space, just evolution of viruses, and then also, so for example, Myosana is a company that is focusing on lipid nanoparticles and trying to basically decorate that nanoparticle with with ligands that will be taken up by the muscle. And then the idea is that, you know, eventually, right now, as the technology stands, it's abysmal compared to AAV, but those approaches are definitely being explored.
Okay, questions from the audience.
Thank you. Hi, I was just wondering whether you thought that in addressing the cost issue of the production, you think it would be more viable to target the manufacturing side or just finding a more effective solution as a whole?
Yeah, so the manufacturing process is, kind of a boring question, but I'm certain that different pharmaceutical companies are in that space. One issue is the ultracentrifugation, but there's also just, you have to produce virus using cells. And then so being able to transfact the cells with the different components that make the packaging process successful, that's also a space that's also being explored by, I wouldn't say exclusively by industry, but industry is more motivated to bring those costs down.
Hi, Dr. Eguchi. It was a very interesting talk. I actually learned a lot. So I guess one of the questions that that emerged as you were giving the talk was I was wondering whether or not there are any, known functional, like qualitative differences in the protein product of the microdystrophins and how they compare to the regular length dystrophin.
You're talking about the protein function.
Yeah.
We know that it localizes to the right place. It goes to the sarcolemma where it's supposed to go. And there's very little information on the structure of the protein just because it's crystal structures are really hard to obtain for dystrophin. There's been parts that have been crystallized, but not the whole thing. And so in terms of function, there are some papers that address specific domains that might be better, it's still up for debate whether recruiting nitric oxide is actually helpful. There's an argument that it's important for the heart, but that evidence seems to be very weak when I surveyed the literature. So it's being marketed that way. But there's still a lot to be explored. There's one paper that discusses how Hinge 2, which is included in the Sarepta product, actually causes problems in the attachment of the nerves, so the neuromuscular junction and the myotendinous junction. And then when you swap Hinge 2 with Hinge 3, then you start to, you basically, all of those problems go away. And so there are certain domains where we know more information, but not every domain has been systematically swapped out like that.
Thank you.
I really enjoyed your talk. Thank you. My question's sort of in the same vein as Dr. Flanagan's question. Are there other candidate viruses that you could potentially use for this, or are they too difficult to work with?
So the beauty of AAV is it's not integrating, so it stays episomal, and so that is what the FDA wants. They don't want non-integrating, because if you have integration that is random and it could potentially drive an oncogene, then that would cause problems. Although, there is an argument for assessing whether a patient is at risk for cancer, but especially because these patients don't live very long. And in terms of different viruses, I mean, all of them have been explored, and then AAV is right now the most efficient way to systemically deliver. So if you're talking about intravenous injection, this is the way to go.
Thank you so much.
Okay, I have one more question. So dystrophin is such an interesting protein because it's so large and it has all these different domains that do these different functions. And so, you mentioned earlier that you're trying to make these smaller ones and hope that the proteins link up with each other. And I was wondering, how far do you think we could push that? Do you think it would be possible eventually to have something where you put in, 5 different pieces or, 10 different pieces, and they have particular sequences at their edges that tell this one to link up to this one, and then the next one to go onto the chain until you can build the whole thing?
Yeah, so it is definitely possible to get 3 different ones to link up together, and but the challenge is getting 3 different components in a cell. And so patients are already getting the borderline toxic dose, and just to get one. And then, so in order to increase the probability of getting 2 different ones to get into one cell, you would have to dose the patient with more. And so right now, that's not possible. But maybe with these more myotropic vectors, we can lower that dose required.
That makes sense. Okay. Other questions? Okay. Let's thank Asuka again.